And this slide is not full screen?
Yeah.
Thank you. Okay, so we are going to continue with the nervous system. So last week we had a very basic introduction into the organization of the nervous system, like the central nervous system versus the peripheral nervous system. And then we talked about how neurons worked at the cellular level with action potentials and dendrites and axons and synapses and all of that stuff. Today we are going to talk on a larger level. It's not just anatomy. There's going to be some physiology involved, but this is primarily going to be anatomy. And we're going to focus on mostly the central nervous system, which is, remember, is the brain and the spinal cord. But we will also talk about the peripheral nervous system. The reason we're going to focus on the central nervous system is because it's much more complex. You can cover the peripheral nervous system in the same depth in a couple slides as it takes 30 slides to cover the central nervous system. So remember, like I just said, the central nervous system is the brain and the spinal cord, period. If it's anything other than the brain or the spinal cord, even if it is attached to the brain or the spinal cord, it is not the central nervous system. And so if you are part of the nervous system, but you're not in the brain or the spinal cord, you are part of the peripheral nervous system. We're going to start with the spinal cord. And so this slide has a list of structures of the spinal cord. These are anatomical features that we can look at the spinal cord and see. And so we're going to start at the top. And we have a cervical enlargement and a lumbar enlargement. So we're going to look at the diagram of the whole spinal cord on the right side of the slide here. And we can see that there is a cervical enlargement right here and the lumbar enlargement right here. And it makes sense if you look at it. The spinal cord is the yellow part running down the center of the spine. And it actually gets a little bit wider here and here. Now, when we talk about the spine, we talk about three different regions. We have the cervical region, which is our neck region. We have the thoracic region, which is our chest, or our upper thorax. And then we have the lumbar region, which is your lower back. That's the small of your back where the spine kind of curves in a little bit. If you have a fancy car and there's a lumbar adjustment in it, you feel that little weird thing come out and poke you in the back, right? That's the lumbar support. That is where the lumbar region is. And we have two enlargements of the spinal cord there, primarily because there's going to be a lot of nerves coming out of the spinal cord in those areas. And so there's just a lot of traffic going through the spinal cord in those areas. And so we have to widen it up a little bit. Think about a highway. When you take multiple highways and merge them together, it's bigger. Eventually you start whittling down lanes, right? Like when the Turnpike and I-75 come together, they're both, I don't remember how many lanes they are, but let's say they're both 3 lanes. All of a sudden they come together and you have 6 lanes, and then it whittles it down to 4. That's why it doesn't have to be thick the whole way. It's just where everything is coming together, it has to be a little bit thicker. And so we have that cervical enlargement and the lumbar enlargement. We have, down below that, we have the conus medullaris, which is right here. And on this diagram, it's not incredibly easy to see if you don't know what you're looking for. But if you look at the spinal cord, start up at the brain and follow it down, and there's a very solid yellow spinal cord. And right when you get to the conus medullaris, notice that everything below that is kind of hairy looking. That's because the solid portion of the spinal cord actually stops at the conus medullaris. And it's called conus because even though they have their little pointer blocking most of it, but it comes down and it forms a little point there. And then the little finger hairs come down below that. The finger little hairs that come down below that is called the cauda equina. And cauda equina is Latin for horse's tail. And so if you look at that, you can kind of see, sure, that looks like a horse's tail that comes off of the conus medullaris. The filum terminale is the very, very bottom tip of the spinal cord. And so there's one of those hairs of the cauda equina that continues straight down. The rest of them kind of curl off a little bit. But there's one of them that continues straight down. In the very tip of that is the filum terminale. The last three terms here refer to regions on a cross-section of the spinal cord. So that's this other diagram down here. Now, we're going to talk about these other things on here. But right now, we're just going to focus on three of them. And I use these three things as kind of my bearings to figure out where am I looking. Because you look at this and you say, well, if I were to ask you, is the front of the spinal cord on the top of that diagram? Or is it on the bottom of that diagram? Now you can look at it and you can see it says the anterior median fissure is at the bottom and the posterior median fissure or the posterior median sulcus is on the top. And so you can figure it out based on that. But on something like a lab exam, that's not going to be filled in for you. And so you need to look at an unlabeled picture of a cross-section of a spinal cord, and you need to be able to figure out where is the anterior and where is the posterior portion. Now, what I want to point out in these two terms, one is a fissure, one is a sulcus. Now, we haven't used these terms yet, but we're going to come back to them on the brain anatomy. A fissure and a sulcus are very similar to each other. They're a valley or a fold. Okay, you can see on both locations there's a fold. But notice that the fissure is larger than the sulcus. And that is always true. A fissure is larger than a sulcus. And the names are anterior median fissure and posterior median sulcus. And so if you see an unlabeled diagram, find these two little folds. Whichever one is the bigger one, that's the anterior or the front side. And by process of elimination, obviously you then know that the other is the back side. The other thing to point out right now is the central canal. There is this hole in the very center of the spinal cord. It runs the entire way up and down, and there's going to be cerebrospinal fluid in there. We'll talk more about that on another slide. So the spinal cord starts all the way up at the top of your spine, at the base of your skull. Now, we haven't talked about the skeleton yet, and so you don't know what the foramen magnum is. But you've probably seen a picture of a skull, and in the base of the skull, there's a big hole. A hole is called a foramen, and magnum just means big. And so this is saying there's a big hole in the skull. That is where the spinal cord is going to start. And it runs all the way from the base of the skull all the way down to the first lumbar vertebra. Like we said, there is cervical, thoracic, and lumbar regions, but there's also a sacral region. That refers to the sacrum, which is a little bone on the back of your pelvis. We'll learn about it when we talk about the skeleton. It looks like a turtle or like a snapping turtle shell. It's really kind of strange looking, but it is essentially your tailbone. That's kind of the back of your pelvis. Your pelvis has two wide sides on it and then a back. The spinal cord is covered with 3 layers of connective tissue. Those layers are called meninges. So that's the plural word. Meninge is the singular form. We have the pia, the arachnoid, and the dura maters. And you need to remember what order they go in. The pia mater is the innermost layer. It is going to be like shrink wrap wrapped around the spinal cord. And then outside of that is the arachnoid mater, and then outside of that is the dura mater. And so I'm going to skip ahead a couple slides, and we can see the 3 different meninges. And so we're looking at the spinal cord. This is a model. This is one of the models in our lab. And we're seeing this out here is a vertebra, so that's the spine. And then right in the center, we have the spinal cord. And there's this white layer going all the way around it. That is the pia mater. Remember I said it's like saran wrap. It's stuck right onto it. Then we have a space. And then we have another layer of white. That is the arachnoid mater. Then we have another space. In this case, that space is filled with adipose tissue. That's this yellow, bubbly looking stuff. That's just fat. And then we have another layer, and this time it's kind of blue, and that is the dura mater. So we learned the 3 meninges, and I showed you the spaces between them. Those spaces have names themselves. And so let's go back to our diagram. So the first one on our list is the epidural space. Epi always means out. And so epidural, let me go back. Epidural. Dural refers to dura mater, and this is outside of the dura mater. And so this is this little space between the blue layer and the bone. So it's very small. I keep hitting the wrong one. Then we have the subdural space. Sub means under. So this is the space under the dura mater. And if it's under the dura mater, that means it is above the arachnoid mater. And so it's between the dura mater and arachnoid mater. So that is this fatty area here. And then we have the subarachnoid space. So this is between the arachnoid mater and the pia mater, which is going to be the open area here. Now, one of these words you've probably heard before. Which one sounds familiar?
Epidural space?
Yes, and why does that sound familiar?
That's when the woman that's going into labor that wants to receive an epidural, that's where they would give the injection, that area.
Yeah, so people talk about getting epidurals. That is a pain block, primarily during pregnancy, but it doesn't have to be during pregnancy. But there's far more people who need or use it for pregnant. There are far more pregnant people than there are people who have other medical emergencies where they require an epidural. Now I want you to look at this figure or this model and look at that epidural space. It is not big, right? Imagine sticking a needle through someone's skin, through the vertebrae, into the epidural space, but not beyond the epidural space. I hope you have a lot of respect for the people that manage to do that. That's a very, very small space with not a lot of wiggle room or room for error. Some other structures associated with the spinal cord are the roots. So this is where the nerves are going to come into and leave the spinal cord. So if I go back, you can see there's these little, they look like Laffy Taffy or something coming out of the spinal cord. Those are nerves. And so where they attach are called the roots. And so where the nerve attaches on the dorsal region or the dorsal side, that is the dorsal root. Where the nerve attaches on the ventral side is the ventral root. Now, on another slide, we had anterior and posterior. Now we're using dorsal and ventral. Do you remember which one is which? I guess if you notice, I guess on my first bullet point, I pointed that out. But the dorsal is the back. Remember, the dorsal fin on a fish or a whale or a dolphin is on their back. And so ventral is on the front. And so we go back to our diagram and we look, the nerve here is attaching to the back. And I know that's the back because it has the sulcus rather than the fissure. It has the small fold in the back. So this is a dorsal root. And so that means this is a ventral root in the front. The dorsal root not only has the root, but also has what we call a root ganglion. A ganglion is a term for a bundle of cell bodies in the peripheral nervous system. So the term ganglion only applies to the peripheral nervous system. We're going to have a different name for the same thing in the central nervous system. Wait. Yeah. No, I started questioning myself because we're talking about the spinal cord, which is the central nervous system. But at this point, this is the nerve that's attached to the spinal cord, and so we are in the peripheral nervous system. I got this. So, that we can't see the ganglion here. Hopefully, here we can see it. So if we follow the dorsal root back, you see there's this big bulb here. That is the dorsal root ganglion. That is where the cell bodies, remember neurons have cell bodies and then very long axons. And so in this ganglion, we are going to have a whole bunch of cell bodies, and then what you see coming from that, attaching to the spinal cord, are axons. That is a single axon from a single neuron that goes all the way from here over to there. That is the dorsal root ganglion. Also notice on this diagram, the dorsal root comes the dorsal root ganglion, it continues on. The ventral root also comes on, and then they get bundled together. Once the dorsal and the ventral roots get bundled together, they are called a spinal nerve. And we'll talk more about the spinal nerves on a later slide. So now let's talk about what is inside of that spinal cord, because there's a lot going on there, and I'm sure you've already noticed that there's two different colors there. We have the kind of yellow butterfly shape in the center, and then we have the white part on the outside. Now that yellow butterfly is generally shown as yellow in models and diagrams, but it is not actually yellow. It is more of a grayish color. The reason they make it yellow in diagrams and figures is because a white and gray figure just doesn't look very good. It's hard to draw a distinction between one region and another. So the butterfly is, like I just said, is normally gray, and so we call that gray matter. The white part on the outside is called white matter. White matter is white because the axons in that area have myelin. Remember that myelin was that lipid insulation that we put on the axons to prevent the electric noise from getting in or getting out, and also allows the saltatory conduction. Remember, we have the oligodendrocytes and the Schwann cells that put down that myelin. Myelin, which is fat, is more or less white in color. And so, in the outer portion of the spinal cord, we have a lot of myelin, and so it looks white. And so we simply call it white matter. In the center, the butterfly portion, where we have gray matter, the neurons don't have myelin. And the natural color of a neuron is gray. And so we can see the gray neurons because they're not covered up with white myelin. The gray matter in the center of the spinal cord is the thinking part of the spinal cord. This is the integrating center. Remember that the term integration means taking sensory inputs and putting them together to create a coherent, essentially, idea. Now, we normally think with our brain, not our spinal cord. But we actually do some thinking with the spinal cord. An example is reflexes. We'll talk about reflexes and learn later on and talk about why reflexes are going to be integrated in the spinal cord rather than further up in the brain. But that is why they don't have myelin, because they are the end of the line. Remember, the myelin is on axons where we carry signals. Neurons that are integrating centers, they're the end of the line. Signals come in, they turn it around and send it back out through motor neurons. They don't need myelin. They're not sending signals anywhere. They're just giving orders. The white matter, though, is carrying information. It's carrying signals up and down the spinal cord. We want that to happen quickly, and so we put myelin on it. That gray matter is the butterfly shape. Parts of that butterfly have different names. We call them horns. There's a posterior or dorsal horn, an anterior or ventral horn, and a lateral horn. The lateral horn is not present in the entire spinal cord. It's only from thoracic 1, and so the beginning or the end of the neck down to the beginning of the lumbar region. That is not a specific range that you need to know. I wouldn't even require you to know that the lateral horn is not present in the whole spinal cord. Okay? You need to know the lateral horn is present in some of the spinal cord, and you need to be able to identify it on a figure. So here we see those horns labeled. And so if you have the butterfly, it looks like there's two wings in the back and two wings in the front. The ones in the back are the posterior horns. The ones in the front are the anterior horns. The lateral horns are more subtle. They are this little bump coming off the anterior horn. They look like it is only just part of the anterior horn, because there are other bumps on the anterior horn that don't have their own special name. The reason we call that its own name is because it's only present in some parts of the spinal cord. The rest of it is there the whole way up, and so it's part of the anterior horn. Rather than saying that the shape of the anterior horn changes as we go up and down, we just say that in some parts of the spinal cord, in basically the middle, there's an extra little lateral horn. Remember, the white matter is the part on the outside. This is going to carry signals up and down. Instead of being horns, we call these columns because they go all the way up and down the spinal cord. We have an anterior, a lateral, and a posterior white column. In each column, we have nerves that run up and nerves that go down. So let me go back. We have the posterior columns in the back, the anterior columns in the front, and the lateral columns on the sides. So you can see that, there's no structure you look at and say, well, that's a column. Just like you see like a butterfly wing. The column borders are more or less defined by the gray matter. And so between the two posterior horns is the posterior columns. Between the two anterior horns is the anterior columns. And then on the sides, between the anterior and posterior columns, are the lateral columns. Now, something that's going to come up, I don't think it's in a slide later on, but it will come up in the lab, is what direction signals run in different parts of the spinal cord. Now, this is not a hard and fast rule, but it is close enough to a hard and fast rule that for this class, you can think of it as a hard and fast rule. That signals in the back of the spinal cord, in the posterior region, are going to move up the spinal cord towards the brain. And in the front of the spinal cord, signals are going to move down away from the brain. The way I remember that is I say back up. In the back of the spinal cord, signals are moving up towards the brain. Now I'm going to ask you a question. If it is moving up towards the brain, do you think those are sensory neurons or are those motor neurons? And while I'm saying this, I just realized you all did the nervous system lab, and so you already know this. So hopefully this is an easy question. Yeah. So a bunch of people are saying sensory. Correct. That actually was probably on your lab exam. Was that just on your lab exam? Anybody remember?
I think that was a question on the lab exam, or I think it was like asked a similar way.
Yeah. I'm saying I think it was there, but I don't remember for certain. So, just a little bit ago, I talked about the spinal nerves. The spinal nerves are where the posterior and the ventral, I just flipped the word. The posterior, no, I had that right. No, dorsal and ventral nerves come together and they continue on. That becomes a spinal nerve. We have 31 of those spinal nerves branching out from our spinal cord as we go down. Some of them are in the cervical region, thoracic, lumbar, sacral, and then there's one coccygeal spinal nerve. I do not require you to know how many spinal nerves we have, how many are in each region. But you need to know that we have spinal nerves, that they run down the entire spinal cord, and that each nerve has both sensory and motor neurons. Because, remember, the nerves come from the dorsal and the ventral and come together to form that nerve. The ones in the dorsal are sensory neurons, the ones in the ventral are motor neurons, and so a spinal nerve has to have both sensory and motor neurons. So that brings us to a nerve. Let's talk more about the nerves. And so at that point, we are leaving the central nervous system, right? We are now in the peripheral nervous system. What does that nerve look like? It is a bundle, essentially a bundle of neurons. And remember, I think last week we learned that a nerve is a bundle of neurons, but also has blood vessels. So that's what we're looking at here in this diagram. This is essentially a bundle of bundles. And so let's start with this. So we're going to start here with a little blue cylinder here. That is a single axon. So that is a single neuron. And notice it has Schwann cells wrapped around it. This is a nerve. It's in the peripheral nervous system, so it is Schwann cells. It is not oligodendrocytes. We then take multiple axons, bundle them together, and wrap them in a coating. That bundle is called a fascicle. So you can see that in the diagram here. And a fascicle is covered by the perineurium. That's connective tissue that's wrapped around that bundle of axons. The endoneurium is inside of a fascicle wrapped around a single axon. So we have an axon wrapped with myelin and then wrapped with endoneurium. We then take a bunch of those, put them together, wrap it up in a fascicle. We then take a bunch of fascicles, throw in some blood vessels, and put another layer wrap around that. That is the epineurium. So the epineurium is going to surround an entire nerve. So endoneurium, perineurium, epineurium. You have to remember which one is which. Epi always means out. So the epineurium is the wrap on the outside. Endo always means in. So it is the innermost layer, in this case, which is around a single axon. And then we have the perineurium, which by process of elimination must be the thing that wraps around one axon. We've talked about sensory and motor neurons. Remember, the sensory neuron is called an afferent neuron. A motor neuron is called an efferent neuron. We can also have a sensory or motor nerve. Now, a spinal nerve is both afferent and efferent, we just said. So that would be a mixed nerve. But once we get away from spinal nerves, so something like in your fingers, that can either be sensory or motor. There are separate nerves carrying signals from the tip of your finger into your spinal cord, and there are separate neurons carrying signals from your spinal cord to your finger, causing it to move. If we look at the spinal nerves coming off of our spine or off of our spinal cord, we see that there are nerves that come off and then branch. A branch coming off of a spinal nerve is called a plexus. And we have 4 primary plexuses in our spinal cord that control different regions. We have the cervical plexus. Cervical means neck. And so this is going to serve regions up around our neck and our head. We move down to the brachial plexus. Remember, the brachial region was your armpit. So the brachial plexus is going to control primarily your arm. The lumbar plexus is going to be nerves coming out controlling the front part of your leg, and then the sacral plexus controls the back or the posterior part of your leg. And I just realized I look like an idiot because I'm showing you the front and the back of my leg, and you can't even tell that I'm doing it, let alone see where I'm pointing. Okay, so we'll do one last slide about the spinal cord, and then we will take our break before we move on to the brain. So I mentioned that the spinal cord has an integrating center. That's the gray matter. And I said that is primarily for reflexes. Now I'm going to pose a question to you and say, why do we want our reflexes controlled by the spinal cord rather than the brain? Because the brain is better at thinking. And so why do we want the spinal cord to do it? There's a reason. Can anybody think of a reason why the spinal cord is better equipped to handle a reflex like pulling your hand off of a hot stove than your brain?
Yeah, I was going to say, just like the guy who messaged that, it maybe has to do with the speed of response.
Exactly, yeah. And so it does not take very long to get from your finger up to your brain. But it takes about twice as long to get from your finger to your brain than it does to get from your finger to your spinal cord. and it takes twice as long to get from your brain back to your hand as it takes to get from your spinal cord to your hand. And so it allows you to react that much faster. In a lot of situations, that wouldn't matter. But in some cases, it does. And not only that, but it allows the brain to focus on higher thought processes. It's not, so that way it's not constantly worrying about, do I have to do this? Do I have to do that? Do I have to do that? Am I burning up? Am I too cold? Is there something flying at my face? Should I wink? Should I duck? What should I do? It can focus on the more complicated things. And so a reflex has a very defined pattern involved. So this is called the reflex arc. A signal is going to go and it's going to come back. So it's going to start with a stimulus. And so we are going to start with the stimulus of putting your hand on a hot stove. And so what do you think that stimulus is? What is the stimulus that's going to happen when we pull our hand off of our hot stove? What's the thing that triggers it? So somebody says pain. What causes pain in this situation? Heat. Yeah. So in this case, the stimulus is heat. A stimulus is something outside of our body. And so this could be heat when the doctor hits you in the little area on your knee. Every semester, I always forget what the name of that thing is. And somebody tells me what it is, and by the next semester, I've already forgotten. Anyways, that little pointy hammer thing that people, I probably become doctors simply so they can hit people on the knee with that little thing. That striking motion would be the stimulus there. If something was flying at your head, the light bouncing off that thing coming into your eye would be the stimulus there. So then we have some sort of a sensory receptor that's going to detect that stimulus. So that is going to be a pain receptor on our hand. That's going to be the touch receptor in our knee. It's going to be the receptors on our retina, whatever it may be. And that is going to send a signal up through a sensory or afferent neuron. Because it's a sensory neuron, it's going to go through the dorsal root ganglion, into the dorsal root of the spinal nerve, into the posterior gray horn, and then the anterior gray horn. And so what really matters is realize it's coming through the dorsal part of the spinal cord into the gray matter. In the gray matter is where the integrating center is going to be. That is what's going to decide. Do we need to pull our hand off of this, or can we leave it there? If we need to pull it back, the spinal cord is going to say, well, I'm a dummy, but I'm smart enough to realize my hand is burning up. We need to pull back. So it is going to send a signal out through the ventral root of the spinal nerve, through the ventral root of the spinal nerve, through a motor neuron to an effector organ. And remember, we used that term effector last week. An effector can be a muscle or a gland. So that is going to be whatever is going to carry out the command sent by the spinal cord. So that would be like the muscles in your arm forcing you to pull your hand back. So we have stimulus, sensory neuron, dorsal root, gray matter, ventral root, motor neuron, effector. That is the reflex arc. You want to remember that pattern. If you can remember that pattern, and make sure it makes sense to you, don't just memorize it, but make sure it makes sense to you. And if it does, you understand how a reflex works. So any questions on nerves or the spinal cord before we take our break? Okay. So it is 6:47. We will come back and restart at 6:55, and so about 8 minutes from now. So it looks like summer is here. I'm looking at the forecast. The highs for the next few days are 93, 95, 96, 95, 95, 94. Yep. The good thing about the cloud, the rain in the afternoon, is it prevents it from feeling that hot. I don't know about you. People always say that it's not the heat, it's the humidity. And I moved here from Michigan, and to me, it's not the heat or the humidity, it's the sun. The sun makes things so much more uncomfortable. When I'm out fishing, to me, the hottest part of the day is like 11 o'clock. Because it's already high 80s, but there's no clouds in the sky, and it's just beating down on you. By the time it's like 3 o'clock, it may be 95 out, but you've got this haze from all the humidity and the clouds, and it doesn't feel nearly as hot at 95 with some clouds and haze as it does with high 80s with bright sunshine. Yeah, so somebody said in Arizona they get to 110. And 110 is still 110. Yeah, 110 is very hot. Then they asked, did I get my boat out of the lake? Yes, I got the boat out of the lake. It was a very complicated process, but I don't remember if I mentioned the lake is surrounded by cypress trees. And so there is no way that you can just pull the boat up on shore. And so it was sunk, it was attached to the dock on one side, holding it up out of the water. The other side was like this, sitting on the bottom. And so I had always thought in my head, what would I do if the boat sank at the dock? And I said I was going to walk out there, pick it up a little bit, put a cinder block underneath it, pick it up again, put another cinder block until it raised it up enough to get it above the water and I could pump the water out. But it turns out there's an unknown depth of muck there. I started climbing off the dock, In the water is only about 2.5, 3 foot deep, but I got up to my neck, and I still wasn't on solid ground. And so walking wasn't an option. And so I broke out my science, and I bought an air mattress, and I floated the air mattress on the water above the boat, put a piece of plywood on top of the air mattress, and then took ratchet straps and hooked one ratchet strap to either side of the boat over the air mattress. And then when I did the ratchet straps, trying to pull the mattress down to the boat, it actually caused the boat to come up off the bottom. And then once it was kind of suspended in the water, we very, very carefully carried it out around the dock to a little bit of an area. We couldn't get it onto shore, but we could get it close enough to shore that the top of the boat was out of the water. Then I pumped the water out of it, and it floated, and there were no leaks. And so it sat there for a couple of days trying to figure out what I was going to do, because this boat that just sank clearly had no leaks. And so eventually, a friend of mine towed it over to the little boat launch area. We put it on the trailer, put a hose in it, filled the dang thing with water, and let it sit and sit and sit and sit. And there was not a leak to be found. And so I'll never know why that boat sank. But today, what I did, I took it into town, and I had somebody weld an aluminum plate over the plug hole, and so there's no longer a plug to leak. I am putting the pump in there. Normally I have to plug the pump in to turn it on. I've set it up so it goes on automatically when the boat starts filling with water, and I'm putting a whole bunch of flotation in there so that if it ever sinks again, it only sinks down to the surface of the water and doesn't go all the way to the bottom. But it's been a process. That's putting it lightly. Yeah, so somebody's saying it's like a lift bag. So my dad he used to be a scuba diver, and he immediately said, well, just hook an airbag to it. You hook a bag to it, then you put an air compressor under it, and it fills with air. And I said, Dad, that's not going to work. The boat is only a foot underwater. The airbag is going to reach the surface before the rope even comes tight. So that's not going to work. But it's the same idea. It's using air to float the thing up. But yeah, I wish it'd be kind of interesting to know how far down that muck goes. But I don't know how I'd ever figure that out. If I take a pole and try to shove it down, there's not enough weight. It sticks. I don't know. But it just kept going and going and going. It's a lot of muck. Okay. So it is time to start again. So we're going to move on to the brain. To me, the brain is more complicated. I mean, it is more complicated than the spinal cord. But learning the brain is tougher. To me, the spinal cord, it just makes sense. You can think of it like a highway, right? We got stuff moving, and we got stuff moving up and stuff moving down. The brain is just kind of a mess. It just signals go in there, and it bounces all around, and magically something comes out the other side. And so you can't think of it as a physical object and like a metaphor. It's not going to work with the brain like it does for the spinal cord. If we look at the brain, there are 4 primary regions. There's the cerebrum, the diencephalon, the brainstem, and the cerebellum. And we'll talk about each one. The cerebrum is what you picture when you picture a brain. So the cerebrum is the outside of the brain, right? When you picture a watermelon, the first thing that you can picture the outside of a watermelon, and you can picture the inside of a watermelon because you've seen the inside of a watermelon. But imagine you had never seen the inside of a watermelon, right? If somebody said, picture a watermelon, you would only picture the outside and you'd say, that's a watermelon. And you would just imagine that the inside was the same as the outside. But when you picture the brain, you're picturing the outside, which is the cerebrum. This is the higher thought region of our brain. There's a number of things it's responsible for, including our senses, communication, memory, understanding, and voluntary movement. So think about it. Most of these things are what separate us from what we would consider lower animals, right? We as humans have better communication than the deer that just walked out my window. We have better memory than that deer that just walked by my window. We understand things better, and we have more voluntary movement. I can sit here and say, I want my arm to go up simply because I want my arm to go up. Now, I don't know for certain that deer don't say that, but I don't think they do. I think most animals have more of a reactionary response. They don't say, well, I want to do this, and so let me do that. The cerebrum is made of two hemispheres. Just like the Earth is made of hemispheres, we have the north and south hemisphere and the east and west hemisphere. We have two hemispheres. We have a left and a right hemisphere. Holding those two hemispheres together is some white matter called the corpus callosum. So this is the cerebrum. If we look at it from the front, we see the left and the right hemisphere. And notice the left and the right are backwards. It is the patient's left and the patient's right. Inside, this dotted line on the left, is the corpus callosum. So this we'll see that again. That's also, I'm going to refer to it when we talk about the diencephalon. But the corpus callosum is going to hold those two hemispheres together. And between the two hemispheres, there's this fold there. That's the longitudinal fissure. And you learned about that in the lab. Now, the cerebrum has both gray and white matter. The brain, if you look at it from the outside, is more or less gray colored, because on the very, very outside of the brain is gray matter. But just underneath the surface is white matter. So here we have a brain where a piece has been taken out. And so you can see the white matter and the gray matter. So you can see that there's this very thin layer of gray matter on the outside, but the bulk of it is going to be white matter. When we look at the cerebrum, we break it down into lobes. Now, our book says there are 4 lobes. That's generally the most common way of looking at it. Some people will show a 5th lobe that's kind of buried down in the brain, but we're not going to talk about that. We're going to act like it doesn't even exist. We're going to say there are 4 lobes of the brain. The easiest one, the one that you should never, ever get wrong, is the frontal lobe. It's in the front of the brain. Think, when you get that question and you get it wrong, you're going to hit yourself right here in the front of your head. That's the frontal lobe. The other easy one is on the sides. These are your temples, right? That is the temporal lobe. There are 2 of them, one on each side. In the back is the occipital lobe. And then in between the occipital lobe and the frontal lobe, going this way, so kind of here, where you would have a bald spot, which I do not have. I can prove it to you. I can turn around, but I don't have a bald spot. But that is where the parietal lobe is. Frontal, occipital, parietal, temporal. Each part of the cerebrum has a number of different functions. Now, what's on this slide is only a tiny little portion of what's going on here, but these are what we consider the most important part. So the frontal lobe is what's going to primarily control our motor function. So when I say I want my arm to go up, It is my frontal lobe sending a signal to my arm to make it go up. There's also the Broca area. The Broca area is in charge of putting out language, and it is only responsible for transmitting language. And so when I'm talking to you, it is what's sending signals to my mouth and my diaphragm to control my breathing, control my mouth shape, to make sounds. It is not actually coming up with what to say to you. It is simply controlling the things that make that language. It is also the thing that would make my hand move to write something. Now, again, it is not deciding what I am going to write. It is simply going to make my hand move in order to write. And interestingly, this is only on one side of the brain. It is on the side of the brain that controls your dominant hand. So I am right-handed. Remember, you're looking at I do know my left from my right. Everything's reversed on the camera. I have a PhD, remember. So I am right-handed. The left side of my brain controls my right hand. For a lot of things, the signals cross. Okay, and we'll talk about why when we talk about the special senses. They'll come back into play with our eyes. But in a lot of cases, the signal crosses. The left side of the brain controls the right side of your body. And so my Broca area is on the left side of my head. One way to remember that the Broca area controls speech or the transmission of language is that in Spanish, boca means mouth. Now, Broca has nothing to do with boca, I don't think, but it sounds a lot like it. And so Broca, mouth, that is the transmission of language. The parietal lobe, so here, is the somatosensory area. These are the senses that are not our special senses. Special senses are eyes, nose, or eyes. Boy, those are organs, not senses. Maybe I don't have a PhD. Sight, smell, taste, things like hearing, things like that. Somatosensories, or somatosenses, are things like touch, pain, temperature, things like that. So that's controlled by the parietal lobe. The temporal lobes control the auditory area, so that's going to interpret sounds coming in through my ears. And it's also going to contain Wernicke area. So that's a German name. You pronounce a W with a V, even though probably 95% of people will pronounce it with a W. It's correctly pronounced with a V sound. So this is what's responsible for understanding language. Now on the slide, it says understanding incoming language, but it is also responsible for understanding outgoing language. And so when you hear me speak, it is going into your Wernicke area, and it is taking a pattern of sounds. I mean, in reality, sound is nothing but air vibrations in the air. That's all it is. And our brain is able to take that pattern of vibrations and convert it into a meaning, which is really crazy when you think about it. But that's happening in your Wernicke area. Now, when I think about what I want to say to you, that also happens in the Wernicke area. I say, I have this idea. The idea doesn't happen in the Wernicke area. But I want to communicate that idea to you. And I have to figure out what pattern of sounds do I need to make so that when you hear it, you will understand what I said. That happens in the Wernicke area. And then what that pattern of sounds then gets sent to the Broca area. And the Broca says, okay, if I'm going to make this pattern of sounds, what does my mouth need to do that? And so the one way I like to think about this, go ahead. Wait, you talk to yourself? So, I honestly don't know. I would guess. Boy, that's pretty weird. I would guess that your other parts of your brain are having this idea. It sends it to your Wernicke area, who then creates that little voice in your head, but then also interprets the voice in your head and sends it back to the rest of your brain, which is kind of a weird. It's like being caught in a loop, right? Which is generally what happens when you talk to yourself. You get caught in a loop and you get nowhere. Right. Okay. So, any other questions? Okay. So, what I was just about to say is one way I think about this is like with a politician. Politicians give speeches. Politicians are very good at giving speeches. Politicians generally aren't very good at writing speeches. They have speechwriters. The speechwriter is like the Wernicke area that says, what do I want to say that is going to convey what I want to say? Then you have the actor, essentially, the politician that goes up on stage and says that. They are the Broca area. They're very good at communicating it, but they're not very good at coming up with what to say. The occipital lobe in the back, its biggest function is controlling vision. Now, I don't have a good reason why vision is controlled back here when your eyes are here. Temporal lobe controls the sound, right? Or interprets sound, which makes sense. Your ears feed right into the temporal region. I think with the occipital in the vision, everything's got to go somewhere, and the front was already taken, and so it went in the back. So this, we talked about this. We don't need to go over that. Again, that's for studying. So we just talked about this. What lobe of the brain is responsible for vision? So one person said D, occipital. Yeah, that's what I was just saying. Vision is controlled in the back. What is the frontal lobe primarily for? Motor function. Parietal up here. Might have to go back a few slides.
There's somatosensory.
Yeah. Parietal was the somatosenses, so like touch and pain, things like that. What was temporal primarily for? What's it right next to? Hearing. Yeah. Okay. So next we're going to zoom in just a little bit on the cerebrum and look at some of the shape of it. If you look at it, we see three primary types of features. We see gyri, or gyri, which is plural for gyrus. We have sulci, which is plural for sulcus. And we have fissures. A gyrus is a ridge, it's a bump. A sulcus is a small groove or a valley between a bump. So let me go ahead a slide and we can see this. So you learned about this in the lab. Remember the top of the mountains here, the hills, the bumps are gyrus, is a gyrus. The little valleys between them are sulci. And then a fissure is a very, very large fold. And so we have the longitudinal fissure between the two hemisphere. Now, if we look at the brain, obviously there are a lot of gyri. But the two that we really focus on are the precentral gyrus and the postcentral gyrus, which are descriptively named because they are right next to what we call the central sulcus, which is this sulcus, this valley that more or less divides the brain in the front and the back. And so the precentral gyrus is the gyrus just in front of the central sulcus. The postcentral gyrus is the gyrus just behind the central sulcus. Now, if I took away the colors on this diagram, it would get much more difficult to pick out the central sulcus. But anytime you are ever going to be asked to find the central sulcus, the precentral or the postcentral gyrus, it'll always have color like this, drawing your eye directly to where you're trying to look. For the sulci, we have the central sulcus. We also have the lateral sulcus, which is here, kind of between the frontal lobe and the temporal lobe. And we have the parieto-occipital sulcus, which is between the parietal lobe and the occipital lobe. For fissures, we have the longitudinal fissure and the transverse fissure. The longitudinal fissure, we said, separated the hemispheres of the brain. The transverse fissure is going to separate the cerebrum from the cerebellum, which is the little baby brain in the back here that we haven't talked about yet, but we will. Now we're going to start moving inside of the cerebrum, inside the brain, and we'll see what structures are in there. So the first thing we have to talk about are ventricles. Now, it's nice to think about the brain as being a solid mass of brain matter, or neurons, or white matter, or gray matter, whatever you want to think of it as. But that's actually not the case. There's a fairly large hollow region inside of our brain filled with cerebrospinal fluid, or CSF. And we have 4 of these. We have the lateral ventricles, the third ventricle, and the fourth ventricle. You already learned about this in the lab, but we're going to cover it again. And so the lateral ventricles are bigger, much bigger than the others. They're called the lateral ventricles because they are on the sides. There's one in the left hemisphere, one in the right hemisphere. And then they both are connected to the third ventricle that is between them. So that is here and here. And then the third ventricle drains down into the fourth ventricle. And the fourth ventricle drains down through the central canal of the spinal cord. Remember, in the middle of that spinal cord there was a hole. And I said that was going to be full of cerebrospinal fluid. That cerebrospinal fluid came from up in your brain. It just then drained down through your spinal cord. Now, I don't remember if I asked you this question in the lab video, but we have a third ventricle and a fourth ventricle. Why do we not have a first and a second ventricle? If we were going to... Yes, exactly. He said they're at the same height and are interchangeable. That's right. So the first and second ventricles are the lateral ventricles, but they're equal to each other. They're tied for first ventricle, and so you're not going to say 1A ventricle and 1B ventricle, so they gave it a different name. Lateral ventricles, and then third ventricle, fourth ventricle. That's very good. So then moving in from the ventricles, we get to what we call the diencephalon. The diencephalon is a very, very complicated region of the brain. There is a lot of stuff that goes on here. When we look at the diencephalon, we break it down into 3 major regions: the thalamus, the hypothalamus, and the epithalamus. The thalamus is going to take signals coming up through the spinal cord, receive them, and then figure out where are they supposed to go in the cerebrum. And so it's going to take one signal, send it to the front, one signal, send it to the back, one signal, and might turn it around and send it back down the spinal cord. So it is the relay center. The hypothalamus is primarily going to control our autonomic nervous system, which we're going to talk about next week. That's our lecture next week. The autonomic nervous system is sort of our lizard brain. It is the most basic life functions, like temperature control, sweating, shivering, hunger, thirst, breathing, heartbeat, everything we need to stay alive in any given time. Something you don't have to think about is going to be controlled by the hypothalamus. And then the epithalamus controls our sleep cycle. It makes melatonin. So here's our diagram of the diencephalon. When I looked for the diencephalon, I used the corpus callosum as my landmark. The corpus callosum is this big white C here. Now the corpus callosum does not actually count as part of the diencephalon. But the diencephalon is everything encased in and below the corpus callosum. So it is this region here. And so the easiest thing to find in the diencephalon is the thalamus. So it is this egg-shaped region here, and it has a little bump on it. In every diagram, in every model you ever look at, the thalamus will have a little bump on it. And the reason that bump is there is because there are two halves of the thalamus, one on each side, and that little bump connects the two. And so when you cut the brain down the middle, you cut through that connecting bit, and so it appears as a little bump, like we see there. So find the bump, figure out what it's on, that's the thalamus. Now, once you find the thalamus, we find the hypothalamus. Think of hypothermia. That is low temperature. So the hypothalamus is below the thalamus. So you found the bump, that means you found the thalamus. Now go down, and that region below the thalamus is the hypothalamus. On the back here, there's this little bulb here. That is the epithalamus, which the epithalamus is the region, but that region is almost completely made up of what's called the pineal gland. And the pineal gland makes melatonin. Melatonin is the hormone that makes you sleepy. It's fairly common for people who might have trouble sleeping to take melatonin supplements when they get the time of day that they want to fall asleep. They take melatonin, it makes them sleepy, and they fall asleep. Normally, the secretion of melatonin is controlled by the rest of the brain. So number one is controlled by the light coming in. If it's light out, it's harder to sleep, right? Because when you have light coming in, it kind of suppresses the secretion of melatonin. When it's dark, melatonin gets made. But we also have kind of the biological clock. Even if we are in a light room, when it gets to be bedtime, we get sleepy. Or even if it's dark, maybe if the power goes out and it's 2 o'clock in the afternoon, even though it got dark, you're not going to get sleepy because it's not bedtime. But when it gets to be bedtime, the melatonin gets made and you get sleepy. And so there's two things controlling it. The light coming in, but also simply what time of day it is. So when you change time zones, the time of day gets all messed up. Your brain thinks it's bedtime, but in reality, it's only 9 o'clock, or I don't know what time your bedtime is. But you should be going to bed. I don't know what Your brain thinks you should be going to bed, but it's not bedtime yet. But after a few days, your brain resets and you get over it. Next is the limbic system. The limbic system is a collection, it's not an area. So it's a collection of structures that work together for a shared function. And so the limbic system is in control of emotion and learning. And so learning is different than memory. I mean, you've had teachers and professors drill that into you for your entire life, right? Learning is different than memory. This is in charge of learning, not memory. So there are three primary parts that function in the limbic system. There's the cingulate gyrus, which is essentially part of the corpus callosum, or it's just above it, it's attached to it. There's the hippocampus, which is down here, and the amygdala, which is here. In this class, you are not going to have to find the hippocampus, the amygdala, or the cingulate gyrus on a diagram. But I expect you to know that those three things are parts of the limbic system, that the limbic system is in control of emotion and learning, and that these structures are in each side of the brain. My Wernicke area can't stop talking to itself about that question. My Wernicke area is going to be talking to itself while I sleep tonight, trying to figure that out. My brain's probably going to explode. So now we're going to keep moving down. We have the brain stem. We have the midbrain, the pons, and the medulla. So if we look at this diagram, we have the cerebrum, we see the corpus callosum, so we have the diencephalon here, and then we have the brainstem here, which essentially connects the brain to our spinal cord. I guess that's all I got for the brainstem. So the pons in the medulla are part of the autonomic nervous system. I don't like the people who made this figure in this book because they just call it the medulla and not the medulla oblongata. And if why in the world would you leave out medulla oblongata? Because then you can make all the Colonel Sanders jokes in the world. So I'm disappointed. So that is the brain stem. And so the last thing, what slide are we on? 33. What's after this? Okay. So we move back to the cerebellum. That is what I like to call the little baby brain back here. It looks like a second brain that is attached to your primary brain. It, like your cerebrum, has two hemispheres. There is a left and right hemisphere. They're connected together by the vermis. So cerebrum hemispheres are connected by the corpus callosum. The cerebellum hemispheres are connected by the vermis. If you cut the cerebellum open, we see these little branches here. And down at the bottom, we can see a little bit of color. And so the branches are made of white matter, and then the rest of the area around it is made of gray matter. And so what we get is this white branching shape. So we call it the arbor vitae. Arbor means tree, like Arbor Day. The cerebellum is responsible for coordination. So it does not send the signal to your legs causing you to walk. But it takes the signals from your frontal lobe, sending signals to your left and right leg, and coordinates them so that they happen at the right time so that you walk. Your frontal lobe is not very good at sending signals at the right time to make everything happen. Like if I reach out and I grab something and then pull it up, there's a lot of coordination involved there. There's a bunch of muscles in my arm that are having to work at the same time at just the right amount to put my arm where I want it to go. That takes coordination. And I need to reach my arm out before I close my hand. If I close my hand and then reach my arm out, that's not going to work very well for picking something up. It is also going to be part of equilibrium and balance, which is what we consider if somebody says someone is uncoordinated. You think of someone that's clumsy, they fall over, they trip, and they also just drop things, they do all kinds of strange things because their cerebellum is not coordinating the signals properly. I really should move these slides around, shouldn't I? So that it's not right after the lecture, right after the slide. So what structure is responsible for coordination? Anybody? Yes, somebody's not off eating dinner. The answer is E, cerebellum. So cerebellum is for coordination. What does the limbic system do? Learning and emotion. We didn't really talk about the midbrain too much. The midbrain is essentially just the connecting link between the brainstem and the thalamus. And the medulla is part of the autonomic nervous system. What time is it? 7:30. So remember when we talked about the spinal cord, we had our meninges. Those were the connective tissue layers wrapped around the spinal cord. We had the pia, arachnoid, and dura mater. Remember DAP? We think I mentioned DAP in the lab video. Starting from the outside, dura, arachnoid, pia. The brain has the same exact three meninges. And not only are they the same name, but they start here and just continue on down the spinal cord. So the pia mater in the spinal cord is the same structure as the pia mater up in our brain. Here we have a little bit different alignment. The pia mater is still like saran wrap, following all the little folds of the cerebrum. It goes down inside the longitudinal fissure and then comes back up the other side. Above that, we have the arachnoid mater, which also goes down into the longitudinal fissure, but is not tight on the cerebrum. And so we have a space between the pia mater and the arachnoid mater, which is the subarachnoid space. Above that, we have the dura mater, the subdural space here between them. But the dura mater, the dura mater does go down in the longitudinal fissure. They all do. The whole thing just wraps around and goes down in the longitudinal fissure. So the deepest meningeal layers. Remember what the word deep means. Which of these is the deepest meningeal layer? Pia mater. Yes. So that means the dura mater is what to the pia mater? What directional term? Superficial. Yes. Thank you for keeping up and answering these. This gets awkward. It's just my Wernicke area talking to itself if nobody answers. So, in those spaces between the meninges, in the ventricles, and in the central sulcus, we have cerebrospinal fluid. And the cerebrospinal fluid is made by the choroid plexuses. There's multiple of these. These plexuses are in the ventricles, the lateral ventricle, the third ventricle, the fourth ventricle, but they are primarily in the lateral ventricles, just simply because the lateral ventricles are much larger, so there's more room for them. This is essentially a filter. That's all it is. We pump blood up into our brain, and we push the blood pressure pushes that blood up against the filter, and the filter lets water through, and little molecules like salt and sugar can get through that filter with the water, but big things like blood cells and proteins stay behind in the blood. So cerebrospinal fluid is essentially just filtered blood. It is more or less blood plasma. That's all it is, and it gets up into our brain and acts as kind of a washing mechanism. We have a lot of stuff going on here. We're constantly remember I mentioned last week all of the energy that goes into running the sodium potassium pump. When you make energy, you make waste products. And so when you make waste products, you need to get rid of them. And so they get put out of the cells into the cerebrospinal fluid, which eventually goes down our spinal cord, gets reabsorbed into the bloodstream, and then goes to our kidneys and our liver to get filtered out. So on this slide, we see the circulatory path of the cerebrospinal fluid. So most of it's going to be made in the ventricles in the very center of our brain. It then comes down out of there. Some of it's going to go down the spinal cord. Some of it is going to go up around the brain. The stuff that goes down the spinal cord, some of it's, like I said, is going to get reabsorbed. Some of it is going to come back up the spinal cord and wash the front of the brain. You do not need to memorize where all these little arrows are going. But you need to realize that most of the cerebrospinal fluid is made in the center of your brain and then has to wash all the way down the spinal cord, around the brainstem, around the cerebellum, and around the cerebrum. So think about all the different brain structures and where that cerebrospinal fluid has to get to. Along with, or similar to the choroid plexus, we have what we call the blood-brain barrier system, or the BBB. This controls what gets from the blood into the brain. We need to get nutrients from our blood into our brain. We need sugar. We need glucose. We need amino acids. We need oxygen. We need carbon dioxide to get out of the brain. And so we need things to move in between the blood and the brain. But we get a lot of things in our blood that we don't want getting into our brain. A lot of things we eat, a lot of toxic things that we eat, that we make in our body, we don't want it getting into our brain. The brain is the most important part of our body, and we need to do everything we can to protect it. And so we have what we call this blood-brain barrier system. So everything that wants to get from the blood into the brain has to pass through this. It's like the bouncer at the door. You have to be on the list to get in. And so there's really only two places where stuff from the blood gets into the brain. There's only two doors. There are the capillaries in the brain, and then there are the capillaries that make up the choroid plexus. Because bad things can get in from the blood directly into the brain, or they can go from the blood into the cerebrospinal fluid and then into the brain. So one type of neuroglia called astrocytes, and astrocytes have a lot of functions. They're kind of a jack of all trades type cell. But one thing they do is they reach little tentacles out and they touch the cells making the blood vessels. So you have a vein or even an artery or a capillary in your brain, and those blood vessels are made up of cells that come together to make a tube, essentially. But remember, when we were learning about cells, we learned there were different types of connections between cells. Some of them left a gap between cells. In blood vessels and the rest of our body, we purposely leave large gaps between the cells that make up our blood vessel so that the blood can very easily flow through. The gaps aren't big enough for the red blood cells and things to leak out. We don't want that happening. But it gives easy access for the water and the nutrients to get out. Here, the astrocytes come along and they create those tight junctions where the two cells are basically welded together. So nothing can get between the cells, meaning that for something to get from the blood into the brain, it has to go through the cells. And so then the cells can control what's going on. It has to go through the door. It can't go through the walls or anything like that. So one thing that is interesting about the blood-brain barrier system is it plays a big role in the design of drugs that we take. If we're treating something in our brain, we need to have a drug that can get into the brain, that can get through the blood-brain barrier system. And most drugs cannot. If you just make a drug without thinking about the blood-brain barrier, most likely it is not going to get into the brain. That is one of the reasons why brain cancer has so few treatment options. Because most of our chemotherapy treatments can't get into the brain. There are very few of them that can. And a lot of the newer, some of the best cancer treatments are made up of antibodies. Antibodies are big proteins, and big proteins absolutely cannot get through the blood-brain barrier system. And so we have this whole class of very promising drugs that are completely irrelevant to treating anything in the brain unless you want to actually inject it directly into the brain, which in most cases we do not want to do. One example of where you might have interacted, or at least run across this, is with antihistamines. It's still kind of spring, right? And it is Florida, so there's always pollen. And so most people probably take antihistamines. When you take an antihistamine for allergies, what do you take? What drug do you take? This is medical information, so you absolutely do not have to share this if you don't want to.
Sorry.
No, fine. Both of those are good.
Sweet.
So I didn't hear what you said.
I said Benadryl.
So you take Benadryl.
I
What-
take Allegra.
Allegra. There's somebody else that said diphenhydramine. Diphenhydramine is Benadryl. Benadryl is the trade name. Diphenhydramine is the generic name. Then there's also somebody that takes Allegra. There's probably somebody that takes Zyrtec or cetirizine or Claritin, which is loratadine. I don't remember. I'm not remembering the Allegra. Maybe fexofenadine, maybe? I don't remember. But is there anybody that takes Benadryl for something other than allergies? Anybody? Or somebody who takes Benadryl. What is a side effect of Benadryl? Yeah. It's a sleep aid. Benadryl makes you sleepy. Benadryl is an antihistamine. It is not something that interacts or is designed to make you sleepy. Getting sleepy is a side effect of Benadryl. But now it's gotten to the point that people will take Benadryl as a sleep aid. It turns out that histamine is a very common little molecule in our body that plays lots of different functions. One of which is allergic reactions, and one of which is in our sleep cycle. Now, allergic reactions happen out in our periphery, not in our brain. Sleep is controlled in our brain. Diphenhydramine or Benadryl can cross the blood-brain barrier. So it inhibits the histamine receptors in our periphery, blocking the allergies, and also inhibits the histamine receptors in our brain, making us sleepy. Now, if you're taking it as an allergy medication, you don't want to be sleepy. And so they came out what we call the second generation antihistamines. That is the Allegra, the Claritin, and the Zyrtec. Those are specifically designed to not be able to cross the blood-brain barrier. And so they block the allergy response, but they don't make you sleepy. So I don't know about you, I find that interesting. That's just a very relatable thing that people I find are aware of, but had no idea what was going on. So next we have the cranial nerve. So just like we have the spinal nerves coming off the spinal cord, we have the cranial nerves coming off of the brain. And we have 12 of these cranial nerves. Now I think in the lab, I told you I do not require you to remember the names for each of the nerves. I want you to know that there are 12 of them. And I want you to know the name of number 10, which is the vagus nerve. And I want you to know the function of the vagus nerve, which is to control your visceral organs. It controls your digestion, your intestines, your stomach. It also plays a role in controlling your diaphragm. And so, most likely, if you ever get a question, whether it's a question I write, whether it's a homework question, whether it's a final exam question, it's asking you about a specific cranial nerve, it is going to be the vagus nerve. Also notice it is spelled V-A-G-U-S. It is not like Las Vegas, V-E-G-A-S. This is spelled differently. You are familiar with your vagus nerve in a couple ways, whether you know it or not. Everybody's gotten the hiccups, right? The hiccups are a spasm of your diaphragm caused by weird signals spiking through the vagus nerve. You get signals that shouldn't be sent. They go down the vagus nerve, they hit your diaphragm, and they cause it to all of a sudden to spasm really hard. What the diaphragm does is it collapses your lungs. And so when the diaphragm collapses or spasms very, very quickly, your lungs collapse very quickly and you hiccup. The same thing happens when you get the wind knocked out of you. Now, most people probably haven't had the wind knocked out of you. Is there anybody here that has had the wind knocked out of you? So one person says yes. What happened, if you want to share, what happened when you had the wind knocked out of you? Could not breathe. Yes. You literally could not breathe. So I instinctively bent over too. Yeah. You can't breathe. And when you can't breathe, you feel like you are dying. And so you kind of hunch over because you can't breathe. The first time it happens, it is really, really scary. It's only happened to me once. And it took me a little bit to figure out what was going on. But even when I did, after I figured out what was going on, it was still really scary. When you are just sitting there or standing there and you try to breathe and you can't breathe, unless you've ever experienced that is not a good feeling. In that case, what's happening is your diaphragm spasmed so much that it spasmed and it stayed there. Your lungs are not permanently, but temporarily squished. And when you try to breathe in, your diaphragm has to relax. It has to come down. And if it's spasmed, it can't relax, it can't come down, and you can't breathe in. Luckily, this doesn't last long enough to kill you. Eventually, it starts to relax, and you get it back into you. But whenever you're watching sports and you see somebody say, "Oh, he just got the wind knocked out of him," get over it. Get up, rub some dirt in it, walk it off, that type of thing. Remember, it is a really scary, really debilitating situation. Another way that you might be familiar with the vagus nerve is the vagus nerve is responsible for at least some diarrhea. Now, you've probably heard of irritable bowel syndrome, or IBS. IBS, it is a syndrome. So a syndrome is just a collection of symptoms that have no singular cause. And so in irritable bowel syndrome, there are a number of things that cause diarrhea when you shouldn't get diarrhea. But one of them is an overactive vagus nerve. The vagus nerve controls the movement of your intestines. It controls the normal movement of food through your intestines. And if you have those spikes coming through the vagus nerve, kind of like your diaphragm spasming, causing you to hiccup, it's going to move things through your intestines faster than they normally should, causing diarrhea. Now, one thing that can trigger this is stress. Stress can cause diarrhea in people that have irritable bowel syndrome. When you get stress, we'll learn about this next week, it triggers the fight or flight response, which causes a number of things. It influences your digestion, and it influences your breathing rate, like your diet, by controlling your diaphragm. It influences your heart rate, all of which are connected to the vagus nerve. And so when you get stressed, the vagus nerve is sending all of these pulses out to the other organs. And in some cases, those pulses make it down to your intestine when they shouldn't. And so that in that case, simply getting stressed can cause diarrhea in someone with irritable bowel syndrome. So somebody said, when you put your face in ice water, the vagus nerve slows your heart rate down. Yeah, so if I never did, I grew up in Michigan, I never did that stupid polar bear plunge where they cut a hole in the ice and jump in. But I did do a little bit of ice fishing, which is horribly boring, which is why I moved here. But if you go ice fishing, you need to learn how to survive if you fall through the ice into the lake. And the first thing they teach you is that when you fall into that cold water, you are going to stop breathing. You're going to hit that cold water, and everything is just going to tense up, and you're going to stop breathing. And if you can't breathe, you can't swim, and you can't pull yourself out of the ice. And so when you fall in, the first thing you have to do is calm yourself down. You have to realize, I'm going to be okay. I need to calm myself down. And when you calm yourself down, in that case, the vagus nerve stops sending that signal. It lets your heart speed back up. It lets you start breathing again. But that's the kind of that fight or flight response when you hit that cold water. So this is a diagram showing where the cranial nerves come from. You do not need to memorize that. So this is not something that you need to memorize for an exam, but which one controls facial expression? So it's a very descriptively named Yes. You said 7th, and all of a sudden I said, oh, crap, now I got to look at the Roman numerals and figure out, is that 7 or not? Yes. That is the facial nerve, number 7. Okay. So that is the end of today's lecture. That's the central nervous system, the peripheral nervous system. Next week we'll talk specifically about the autonomic nervous system. If you have any questions about the lecture, about the class, anything before we sign off? Okay. So, if not, I will see you next week. You already did the nervous system lab, and so there's really no lab for you to do this week, and so you've got a little bit of a break. Breaks are always good. Okay, so If you have any questions, let me know. Otherwise, I will see you next Tuesday night. Have a good night.